Hello children. So today, as I told you, we are going to learn about the importance of sunlight in the photosynthesis process. And in my previous video, we are seeing how chlorophyll and carbon dioxide are very important for the photosynthesis process. So today we'll see the essentiality of the sunlight. Now here, as you see, again I've kept this potted plant here. So before we carry out the experiment, so what did you do? We have to keep this potted plant in a dark room, okay, that is for 4 to 5 days. And when you keep the potted plant for 4 to 5 days, what is the process happening there? That is, the de-starching will take place. So I told you, during that process, when you keep in dark room, no sunlight will fall on this potted plant, and all the food which has been stored in this plant in the form of starch will be used by the plant for carrying out the metabolic activities. And that is how after 4 or 5 days, all the starch will be used up. So that process is called as de-starching. And after 5 days, we have to take out this potted plant and keep it in sunlight. Before we keep it in sunlight, you have to select one of the leaf, okay? And this leaf, you have to cover it like this. The middle part of the leaf, both upper side and the lower side, you have to cover it with this black paper in this manner, okay? And then you shouldn't pluck it, okay? Don't pluck the leaf. You should be with the plant only. So you have to attach it with the help of the pins or paper clips, okay? With the help of paper clips or with the help of stapler pins, you can fix this paper to this leaf in this manner. Don't pluck it, okay? Then keep this arrangement for 5 to 6 hours in the sunlight, okay? So you have to keep this potted plant in the sunlight for 5 to 6 hours. And after 6 hours, now you can remove this paper and then you have to pluck this leaf, okay? The selected leaf which you have taken, this you have to pluck it out. And we have to carry out the same experiment what we did in the previous 2 experiments. So once again, let us see what are the steps we have done in the previous experiments. So I told you the first step in the experiment is to take a beaker containing water and we have to boil the water. In the boiling water, we have to add the leaf which we have selected. So this selected leaf which we have just now plucked, so that leaf we have to put in this boiling water. And for 5 to 10 minutes, you have to boil it in this boiling water. And what is the main purpose of boiling it in water? So that the living cells of the leaf all will be killed. So this is how the leaf is been killed. And then after 5 to 10 minutes, with the help of the forceps, you can remove this leaf from this boiled water and then now place it in another beaker containing the alcohol, that is the ethyl alcohol, okay? And this beaker, you have to place it in the water, that is the hot water, okay? So you have to indirectly heat this ethyl alcohol, okay? So when you are indirectly heating this alcohol, so the main purpose of heating alcohol is to decolorize it. That means all the green pigments will be removed from the leaf, okay? Decoloration. So the whole leaf will turn pale in color and the water, all the pigments, the green color pigments, that is the chlorophyll pigments, will be removed from the leaf and the leaf will become pale in color. And once all the color has been removed and the leaf has turned pale color, now we have to remove this leaf and thoroughly wash it in cold water. So this is how, after washing it, then it is ready. The leaf is now ready for the starch test, okay? Now for the starch test, we have to take the iodine solution, okay? So what you do it, you apply 2-3 drops of iodine solution on the whole leaf, okay? So you apply the iodine solution, dilute iodine solution on the whole leaf and wait for few minutes. And after few minutes, what you see is, you will see there will be a change in the color. So this is how, where the portion which was covered with the black paper, that remained unchanged. There was no change in color. Whereas the portion which was exposed to the sunlight, okay, that area change into blue black in color. Okay, so that area, the area which was exposed to the sunlight, after adding the iodine solution, change into bluish black in color, whereas the area which was not exposed to the sunlight, which was covered with the black color, it remained unchanged. The reason is the area which was exposed to sunlight carried out photosynthesis process, and the food was prepared here in the form of the carbohydrates, and which was stored in the form of starch. And that's why when you added the iodine solution, it showed the positive test towards the starch test. Whereas in this area, as it was covered by the black paper, the sunlight couldn't reach there, and because of that, the sunlight was not trapped in this particular area because it was covered by black paper, and because of that there was no photosynthesis carried out in this particular area. And when we add the iodine solution, there was no change in color. Okay, so I hope you understood from this experiment that sunlight is very, very important for the photosynthesis process. So from all these three experiments, we come to know that carbon dioxide. Okay, so from all these three experiments, what we have studied, that is carbon dioxide, sunlight, and chlorophyll. All these three are very important for the photosynthesis process, and any one of these, if it is absent, photosynthesis process cannot take place. Okay, all the three are very, very important. All the three are very, very important. Photosynthesis process cannot take place. Okay, so I hope you understood. Carbon dioxide, sunlight, and chlorophyll are very, very important for the photosynthesis process. Now, not only these three are important for the plants for photosynthesis, but there are other things which also are important for the plant growth. Okay, plant not only requires these things, but for the complete growth, healthy growth of the plant, the other essentials which are very, very important. The first and the foremost is the water, and we know the water is absorbed through the soil with the help of the roots, and that is how it reaches to the whole plant. So water is absorbed from the soil, and with the help of the roots, it is supplied throughout the plant. And the other important things which are very, very important are the macronutrients and the micronutrients. Okay, macronutrients and the micronutrients. So these are very, very important again for the plant growth. In 9th standard you have studied about macronutrients and micronutrients. Some of them are, you have studied about nitrogen cycle, nitrogen, phosphorus, iron, magnesium, and so on. Okay, so in 9th you have studied about nitrogen cycle, where you have seen the nitrogen is taken in the form of inorganic form, that is in the form of nitrates and nitrites. Okay, so this I have already explained you how nitrogen is been converted to nitrates and nitrites, and how it has been absorbed by the plant in the inorganic form, whereas some of the nitrogen is also been converted in nitrate and nitrites by biological process fixation. Okay, some of the bacteria also help in fixation of nitrogen. Okay, and that is converted to nitrates and nitrites, and that is how plant will absorb it in inorganic form. All this we have studied in 9th standard in the nitrogen cycle. Similarly, phosphorus, iron, magnesium, all these also are absorbed in the form of inorganic material. They dissolve in water, and that is how these nutrients enter into the plant and they help in the plant growth. And whereas nitrogen is mainly used for the preparation, synthesis of proteins and for the other metabolic activities. And this is how the plant will grow in a healthy plant. So any one of these, if it is lacking, plant will not have a healthy growth. So here we have completed the autotrophic mode of nutrition. So children, here we complete the autotrophic mode of nutrition. So I hope till here whatever topics we have covered in this particular section of autotrophic mode of nutrition, I hope you have understood everything in detail. Okay, so in my next video we will start with heterotrophic mode of nutrition, where we will study the different types of heterotrophic mode of nutrition. Then we will see the holozoic mode of amoeba, and we will also study how the digestion in the human being takes place. So for time being, I think you have all understood. So you are studying. So have a nice day. Goodbye.